Okay, welcome back, and sorry for this technical issue. We've heard from Laurent the historical development of the disease from a clinical point of view, and in trying to substitute for Pier Paolo Pandolfi, who couldn't make it, I decided I would just review with you 30 years of mechanistic studies to try to understand really how we could provide a rationale behind the miracle, and how understanding this miracle could actually lay the ground for future types of approaches. And so you've heard again from Laurent the in vivo differentiation, complete remission, but remissions are transient. I think this is important to state. And the combination of retinoic acid and classical anthracycline-based chemotherapy initiated here in France by Laurent de Goos allows now 70% cures. And so this, of course, was associated with the 1517 translocation, the PML-RER fusion. Retinoic acid receptor is a nuclear receptor responsive to the hormone. PML, as we'll see, is an organizer for nuclear domains which actually play a very important role in the pathogenesis of the disease, and even more so in the basis of therapy response. One of the things that probably made APL so simple to dissect mechanistically is that APL is a rare example of a cancer which is monogenic. And there is very good epidemiological evidence that the incidence of APL is flat with age, pointing to a single rate-limiting step, and genetic studies that have explored the APL genome in some details have shown that some cases actually only have PML-RER as a single mutation and nothing else, at least in the exome studies. And you have cooperating mutations such as FLT3 activation, which is relatively common, but which is obviously a progression step and not to the same level than PML-RER. And retinoic acid, of course, binds to the RER moiety of PML-RER, and therefore this was the first example of targeted therapy wherein the treatment directly acts on the oncogenic protein. And so a model was very rapidly proposed, which is really a model for transcription differentiation-based therapy, proposing that the PML-RER complex, when bound to DNA, is a powerful transcriptional repressor, and that this repression is responsible for the differentiation arrest. And in the presence of retinoic acid, then you will reactivate target genes, and this target gene reactivation is responsible for overpassing the differentiation block and restoring differentiation. And these cells, of course, are very short-lived and will disappear. And so essentially in the early 90s, it was believed that the story was over, that we understood everything, and that there should be something else. But when you look into the details, and in science you should always look into the details, some things did not fit. And among the things that did not fit is that the concentration of retinoic acid that must be provided to patients was way over the KD of binding of the receptor. You need really to sustain very high concentrations to get clinical effects. And as soon as you go down, you will lose the clinical efficacy. And over the years, actually, a number of observations came in to question this model. And needless to say that because this model was simple to understand, it made it to the textbooks immediately. And then it was quite difficult, actually, to start raising issues. It's always harder to break an established model than to propose one. And something that Laurent de Goos alluded to is that it was unclear why the synthetic retinoids that were used to treat patients in the early 80s in Europe, Japan, and the United States. In fact, when you look very carefully at the case records, there was differentiation. So the leukemic cells did differentiate, but there was no clinical response. And these drugs were abandoned, of course, because they were clinically useless. Then I'll show you some examples where the first example of genetically defined mouse models of APL were derived. Some of them, and I'll show you an example, did exhibit terminal differentiation, yet there was no tumor response. So in these cases, we could really completely uncouple on a genetic basis differentiation response from the anti-proliferative response. As I mentioned, the loss of self-renewal, in fact, in a number of settings, requires extremely high dose retinoic acid concentrations. And finally, we stumbled upon what we called uncoupled retinoids, which are in fact correspond largely to those synthetic retinoids initially used in the 80s, in which we can completely uncouple differentiation from self-renewal, and I'll show you also some example of that. And finally, the last piece of evidence that convinced the group that differentiation and anti-leukemic effect were not absolutely superposed concepts is that in ex vivo transformed cells, the differentiation triggered even by all trans retinoic acid seems to be fully reversible. And actually, I'll show you some recent data for that. So some illustrations of these concepts. Genetically uncoupling, well, this was done using the variant translocations. PML-RER represents 98% of the translocation cases, but there are rare variants, so-called variant translocations between chromosome 11 and 17 that yield the fusion of a very important stem cell gene, PLZF, with RER. And these PLZF-RER leukemia are known to be somehow clinically resistant to retinoic acid, but when we looked at the mouse models that were generated by Pier Paolo Pandolfi, it was very clear that you had full differentiation, and that this full differentiation was not accompanied in any way with tumor regression. So in this case, we had in vivo differentiation, but this was essentially converting an undifferentiated leukemia into a differentiated leukemia, but this was not accompanied in any way by tumor regression, even with very long treatments. Another piece of evidence gathered at the same time was just to do a basic clonogenic assay in vivo. So we take an APL mouse, we treat them, we collect the leukemic bone marrow, and we re-inject the leukemic bone marrow into a secondary recipient, and then we have a dramatic dose response between the very high doses, with those which are required to cure the patients, and the more intermediate doses, although they induce the same level of morphological antigenically differentiation, you have a dramatic effect on the clonogenic elimination of clonogenic cells, pointing us again to the fact that doses are extremely important, and raising also some explanations on some Chinese trials, which were already de-escalation trials, in which the doses of retinoic acid used in the patients had been decreased, and this was accompanied actually by a greatly increased incidence of relapses. Moving now to the uncoupled retinoids, again, we stumbled across on this largely by chance, comparing the classical all-trans retinoic acid and synthetic retinoids that popped out of some screens. When you look at gene activation, you have identical ability of these synthetic retinoids to target, to activate primary targets. When now you look at their ability to degrade the PML-RER fusion protein, you see that they do not degrade at all, although we have full differentiation. And when you look at the clonogenic activity of those treated cells, you see that despite complete differentiation, only retinoic acid will abolish the ability of these cells to create colonies in methylcellulose. And this was quite a striking example of the uncoupling between these two phenomena, but perhaps the most dramatic experiment was the following, performed in vivo in mouse models. So we have leukemic cells treated with retinoic acid. We get granulocytes fully differentiated of leukemic origin, and if you re-inject them into secondary host, they will never reinitiate leukemia, so these cells are no longer immortal. But if you take now cells treated with uncoupled retinoids, you have fully differentiated cells with the remaining PML-RER expression, and these cells actually can de-differentiate in vivo and reinitiate leukemia, again, pointing to the dissociation between differentiation and loss of clonogenic activity. And along the same lines, studying ex vivo models of transformation, wherein progenitors are transformed by expression of PML-RER, they will yield colonies which are immortal, and should be stated that actually in this system, only perhaps 1 in 100 cell has clonogenic activity. So we really have a recapitulation of the stem cell model. Despite the fact that all these cells are fully undifferentiated, only 1 in 100 will be clonogenic and will have the ability to reiterate clonogenic growth. And if you treat with retinoic acid, you have a dramatic decrease in the number of colonies, you have full differentiation. Everyone had believed, including myself, that this would be a full stop. But if you wash away the retinoic acid and replate these cells into fresh medium, they will regrow, and they will regrow essentially at the same clonogenic activity. And so this was carefully quantified, and we came to the conclusion that in the basal states, we have in these primary cells only 1% of clonogenic progenitors, the rest being promyelocyte-like cells. And if we treat with retinoic acid, we'll have essentially all of the cells will be differentiated, but a fraction, not so different from the initial one, will remain clonogenic and will be able to reinitiate the disease. So this led to the idea that we had 2 different uncoupled phenomena, differentiation and loss of clonogenic activity. And just to comment on this model, a paper by the group of Ross Dickens, which is going to appear in the coming weeks in cell stem cell, really established the same concept now, not in the case of APL, but in the case of AML, using a genetically constructed AML model, wherein you have p53 null cells in which you can manipulate the level of the master gene PU1. This PU1, which can be switched on and off by doxycycline, will induce, if you shut it off, it will induce the differentiation block and the leukemic state. If you restore it, you can, of course, differentiate these leukemic cells fully to the point where they get to be granulocytes. And then if you suppress in these granulocytes PU1 expression, you can actually very efficiently revert to the leukemic stage, providing a striking example, a striking similarity with the APL system, wherein retinoic acid will allow the shuttling between these 2 stages, and wherein this fully differentiated cell is not irreversibly gone to the dustbin, but in some conditions can actually move back towards the clonogenic state. And this actually is a perfect illustration of the concept that we developed in a recent review article. So we had a problem. We had a problem that we knew that there were two different pathways activated, but we didn't know exactly what they were. And the answer really came from the exploration of the arsenic model. Because, as mentioned by Laurent de Gauss, arsenic is another drug that has essentially the same ability to trigger differentiation in vivo. These are images from patients actually taken from Saint Louis. Again, similar to retinoic acid, we have 95% complete remissions due to this drug. We have some in vivo differentiation, but strikingly, and in sharp contrast to what I told you for retinoic acid has a transient therapeutic benefit as a single agent, but arsenic can cure 70% of patients as a single agent. So despite the fact it's it's triggering differentiation, but this implied that it was doing something else, and that the something else that it was doing was critical to its therapeutic efficacy. And together with Chen Su, we worked extensively on this model because also it was very clearly incompatible with the old transcription model because retinoic acid, arsenic does not affect retinoic acid receptor signaling, and therefore there was something else that was to be discovered. And what was discovered is that actually the two drugs have the ability to target PML-RER degradation, and it targets PML-RER degradation by direct binding to retinoic acid receptor or to PML for the arsenic part, and the biochemical pathways were largely described in the lab by Junsu and Valérie Lallemand. And so this actually shed a new light on the other facet of PML-RER dependent biology, which is the ability to disrupt PML nuclear bodies. And so these PML nuclear bodies, which you can see here, are scaffolding structures that will recruit and promote the post-translational modification of a very large number of partner proteins. And among the effects that it has, that is very well documented in the literature, is that these PML nuclear bodies will control senescence induction, at least in part through the activation of P53 signaling and also the deactivation of cell cycle through a direct effect on E2F. And in PML-RER, PML-RER has the ability to disrupt these PML nuclear bodies, and that you can see here very nicely. These are normal cells, these are leukemic cells, and so the effect through direct contact of PML-RER expression is to deorganize those PML nuclear bodies, which of course will blunt P53 signaling, and this blunting of P53 signaling was clearly associated, of course, to the pathogenesis of the disease. And as you can imagine from the Western blot I showed you 2 slides before, if you degrade PML-RER, you will restore very rapidly the formation of nuclear bodies in vivo. So these are, again, historical slides from Marie-Thérèse Daniel in patients treated in Saint-Louis with retinoic acid. These are blasts in both cases. This is the untreated form. You have these microspeckled pattern of PML nuclear bodies. And if you treat with retinoic acid, these blasts will have, despite the fact that they remain undifferentiated, they will have reformed the PML nuclear bodies. And exactly the same is observed for arsenic therapy, where the restoration of PML bodies is immediate. And so this had suggested from the start that there may be a role of PML nuclear bodies in clinical response, and we'll see that actually this role was very clearly shown in mouse models to promote the loss of self-renewal. And so this was primarily the work of Julien Ablin during his PhD. He is in the audience, and to show that PML-RER has these 2 facets. The facet of transcriptional repression, which was the first one to be discovered, which largely explains the differentiation block, and the pattern of nuclear body deorganization, which probably bypasses senescence mechanisms. And when you treat with retinoic acid, you will activate transcription, resulting in differentiation, and you will also degrade PML-RER, allowing the reformation of these PML nuclear bodies, activation of P53, and loss of self-renewal. And really, these are the 2 facets of therapy response, differentiation and loss of self-renewal. And these 2 facets are triggered at different efficacy by each of the 2 drugs. In the case of arsenic, we have exactly the same. We have actually PML-RER degradation, which will free those bound promoters from their repressor sites, and allow the triggering of passive differentiation in a way, and the direct reformation of PML nuclear bodies reactivating P53. And so what we can see in the mice, really to illustrate this, is that, of course, one of the key was to generate APLs devoid of the normal PML protein, and these APLs without PML actually will induce some differentiation, but no arrest of self-renewal and no activation of P53. You can see here with retinoic acid, you have a clear activation of P53 very early after therapy, 12 hours in vivo, which is really concomitant with the first wave of transcriptional activation, and you have nothing in the PML nulls, you have no clinical response as well. And if you look with arsenic, it's even more drastic. You have a dramatic stabilization of P53 only in the presence of PML, arguing that genetically PML is upstream of P53 and controls its stabilization. And indeed, again, in mouse models, although in P53 plus or P53 minus APL's differentiation is identical, the ability to clear leukemia-initiating cells is drastically affected. And in the case of the wild-type animals, we have very clear disappearance of the leukemia-initiating cells, and this clearance is not observed at all when P53 is deleted, despite similar induction of differentiation. So this led to, of course, the question of whether the two drugs should be combined or not. And taking differentiation as an endpoint, it was very clear that there was actually an antagonism of the two drugs for differentiation, which for a long time precluded their combination in patients. And it's going towards animal models that showed that there was actually a dramatic synergy for the clearance of the disease. This synergy, of course, reflecting the synergy for the elimination of self-renewal. And you can see that as soon as the leukemia does not self-renew and differentiate, you have an incredibly rapid clearance of the disease. This is less than a week in vivo, and you can see that you can almost eradicate the disease. You can actually eradicate the disease in a week. And this is accompanied by prolonged survival, as shown by prolongation of survival in treated animals with single treatment, elimination of the disease in the dual treatment. And this led, again, to clinical trials, which have perfectly mimicked the findings made in the animal models, allowing the elimination of the disease in virtually all patients. This was initially in the favorable risk patients, but as Laurent de Gosse mentioned, now this has been extended to all patients, including the high-risk patients, where the disease can be rapidly eliminated. And again, I think it's important to stress that patients will get off treatment, so this is not a continuous anti-kinase addiction. Here, the clone is really eliminated definitively, and you can get oral therapy now with oral sulfur. So for a long time, we believed, here again, I mean, the physiopathology was really based on the in vivo findings. For a long time, we believed that the basis for this synergy was simply that the degradation of the protein was accelerated, and it made full sense because the degrons are different. You have an N-terminal degron, a C-terminal degron. If you put the two drugs, the degradation of the fusion protein will be much faster, and you can see this in vivo. This is only 6 hours, so this is really contemporary with the first wave of transcriptional activation. You have a full reformation of these nuclear bodies. So synergistic degradation for a long time was the preferred model, and Chen Su and I wrote an alarming number of reviews saying that we were just essentially treating better because we degraded better. But then the later finding that normal PML is absolutely required for disease elimination raised another issue, which is that arsenic directly acts on normal PML. And therefore, not only retinoic acid and arsenic would induce the synergistic degradation of the fusion protein, allowing the passive reformation of nuclear bodies, but actually arsenic, by acting directly on the normal allele of PML, also could accelerate the reformation of PML nuclear bodies and enhance the activation of P53. And you've seen that the stabilization of P53 in vivo is much higher with arsenic than with retinoic acid, speaking to the possible relevance of this model. And this, of course, could explain very well, this model could explain very well the potency of arsenic, and remind you that retinoic acid alone is almost never curative, whereas arsenic alone can be curative in the majority of patients. Perhaps talking to this dual and probably unique to my knowledge model, wherein the same drug actually will destroy the oncogene and activate the anti-oncogene through reformation of PML bodies. And we were really delighted again to get some feedback from patient studies showing that in historical patients who had not benefited from frontline retinoic acid arsenic therapy, resistance did develop. As mentioned by Laurent de Goos, retinoic acid resistance was linked to mutations in the ligand-binding part of PML-RBR. This is perfectly logical. In most cases, this abolishes ligand binding and degradation. Arsenic resistance was linked to highly clustered mutations in the arsenic-binding moiety of PML-RBR, which we had identified as a D-cysteine motif in one of the zinc finger domains. D-cysteines are the preferred binding sites for arsenic atoms. But we were surprised to discover in one resistant patient, actually, the same hotspot mutation on the arsenic binding site, but on the normal allele of PML. And the discovery of this mutation on the normal allele of PML, which we first reported, but was then also confirmed by other groups, actually genetically establishes these two arms. The fact that arsenic will act both on PML-RBR and also on PML, and that both actions of arsenic are critical to the therapeutic response. Okay, so what are the lessons that can be taught from APL? One of them is that differentiation of leukemic cells can be achieved in vivo, and I'm sure that Parrish will come back to that in his talk in a second. Differentiation on its own has a major debulking activity, but this activity of debulking remains transient if there is not also a self-renewal signal, and this anti-self-renewal signal can be provided either by very high doses of retinoic acid or by combination with chemotherapy, which in this case, in the historical patients, would activate the self-renewal, or by arsenic. Also, degradation therapies are feasible, and I can tell you that when more than 20 years ago we proposed that degradation of PML-RBR by arsenic was the basis for its response, these proposals were not welcomed by the community. Even some people claimed that it was simply obscene to think that protein degradation could underlie biological response, because in those days, transcription was everything, and if you did not modulate transcription, you would have not a chance of doing anything. And now, of course, these multiple farmers now have engaged into massive degradation screens for many, many types of drugs. Also, the good news is that targeted therapies can be curative, particularly in association. And if we can pull out the right association, then you can get elimination of the disease. And I'm sure that several of the upcoming talks will address this question. So, differentiation was transcriptional control. What is control of self-renewal? Well, in the case of APL, very clearly, the basis for self-renewal is the control of a PML p53 senescence axis, which can be promoted by arsenic. And it's absolutely required for APL cure, and some of our current studies at the moment are really trying to see whether this PML p53 axis that we have identified in APL, how can we model its function, what are the determinants, how does PML activate p53, which remains a very broad mystery, and also, well, you know, what is curative in one disease may be curative in another, and so are there other conditions in which we can activate the same pathway for therapeutic benefit? The other one is that synergistic drug combination based on physiopathology and not trial and errors approach should be thought after an in vivo model, and I'm sure we'll hear from this also in Cedric and Frédéric's talk on the rationality of combinations in the case of hedgehog signaling. And also, coming back to the PML p53 senescence axis, can we have PML-directed therapies in other malignancies? And for this, I'm sure we'll have insights from Ali's talks on the role of PML in other conditions. And so the hope is that, as mentioned by Laurent de Goos, by studying the miracle, maybe the miracle will no longer be a miracle, but the miracle will become a model. It will become a model for other conditions. And so what is our current approach, which was just supported by an ERC grant to the group, is that possibly PML nucleobodies constitute a central mechanism in the control of self-renewal by allowing the activation of senescence in a number of situations. I think we established it pretty clearly in the case of APL, but there are hints that this may be a very general mechanism, and that this PML P53 senescence cascade can be actually therapeutically manipulated in a number of other conditions. And we're looking for some of these conditions, and actually identifying those conditions came in part through the study of interferon, because PML gene is massively controlled by interferon signaling, and interferon signaling will also activate the formation of the expression of the SUMO modifiers. And the combination of interferon to massively upregulate PML and arsenic to promote the aggregation of PML nuclear bodies will allow the formation of these domains, which we envision, and Valérie Lallemand has now very definitive examples of this, validation in vivo. We envision this really as a catalytic chamber that, in response to stress, will allow the post-translational modification of a broad variety of proteins, resulting in a number of responses, one of these responses being induction of senescence. And this actually is very easy to manipulate in vivo. This is just illustration of mouse livers untreated. You see rare PML bodies, and if you treat with a combination of retinoic poly-DIC, which will induce interferon and arsenic, you have this massive occurrence of PML nuclear bodies, which will in fact induce a SUMO storm and induce a massive sumolation of a very large number of proteins. And we think that this can be used as a tool to fight at least some malignancies, and particularly those malignancies which were previously reported to be somehow sensitive to interferon, and we have some evidence that this sensitivity to interferon actually reflects activation of this pathway. And in closing, I'd like to thank my many collaborators, including here in the audience, John Zhu, Valérie Lallemand, and Julien Ablin, who is here, and thank you all for your attention.